Yes, sure, we can see your screen.
Okay, good. Thank you. So today, as the title goes, progress in biomapping activities in Nigeria. So we'll be talking about biomapping activities, and we'll see the definition as we go into the presentations. But of course, we'll be dealing with the photographs and act of photography or wildlife photography in general. Okay, so what is biodiversity mapping? There are several definitions for biodiversity mapping, but it is actually the skills of observing natural phenomena, identifying different species of organisms, ranging from reptiles to amphibians to mammals, birds, insects of different varieties, forms, and classifying them into categories, either families, classes, genus, and others, and mapping the data for conservation and management. It can also be defined as mapping of species and eyeing on their presence or absence or count, that is in terms of abundance number, within an area at a specific time or location, which in other words can be defined as richness, that is the number of species found in a defined area or region or locality. So these maps can reveal patterns between a specific landscape, maybe forest, agricultural landscapes, or urban landscapes, and the organisms that live there. And these maps you see here is the number of species, a biodiversity map for the number of amphibian species richness, according to Gallant et al. 2007. You can see where we have the greatest species richness for amphibians in the Southern Africa. There are over 600, more than 640 species. Then of course, in Northern Africa, if you look at the map, you see that the number of amphibians is 1 to 10. And that is not surprising because this is a landscape dominated by the Sahara. And if you look at the Arctic region, the same scenario. If you look at Australia, the same scenario. So that is basically biodiversity maps or biodiversity mapping, and we'll be seeing most of this as we go into the presentation today. More on definitions, biodiversity maps illustrate the extent of impacts when variables relating to anthropogenic pressures are integrated with biodiversity data. Apart from mapping the presence or count of biodiversity in a given location or at a global level, when we relate some of these biodiversity maps with anthropogenic pressures, anthropogenic variables, or any other environmental variables, let's say like climate, maybe like temperature, we can see patterns of biodiversity distributions across the larger landscape. Environment and biodiversity just questions, we can answer some of the interesting questions about some of these anthropogenic pressures on biodiversity or environmental impact of biodiversity. Today, we are talking about global climate change. Species are shifting their ranges, or their ranges is constricting, or maybe in the case of migrants, they are delaying their migratory their migration, or they are advancing their migration. So we can talk of we can answer some of these questions when we relate biodiversity maps with anthropogenic data or environmental variables. And this map is biodiversity maps for the biodiversity loss in terms of percentage compared to an intact ecosystem where there is high human activities and areas with low human activities. You can see in Australia, there is more than 40% of biodiversity loss. And that is also not surprising because of the frequent wildfires in Australia. If you look at sub-Saharan Africa, you can see it's also red. That is more than 40%. If you look at Southern Africa and parts of Asia and also parts of the United States in North America, you can also see the patterns of biodiversity loss. So these are some of the interesting things we can answer, we can see with biodiversity maps. These maps are a very useful tool in identifying important locations for biodiversity agenda, because when they are mapped, we can pinpoint areas of biodiversity hotspots. In the case of Conservation International, we can identify areas with high biological diversity. especially when data collection requires collaborating with others. Because for the Conservation International to identify areas with high biological diversity hotspots, there is need for that data to be available to make an informed decision. Yeah, we can identify this hotspot as biodiversity hotspot, or we can identify this area as a Ramsar site or as an important or biodiversity area, or as a freshwater key biodiversity area. You need this data for us to make an informed decision about protected area management or identification, especially collaborating with others, maybe be at the national level or at international level. Identify the range of occurrence of harmful species, like invasive species that poses serious threat to both biodiversity and humans. Also, through biodiversity mapping, we can identify the extent of occurrence or distribution of invasive species. And invasive species are those species that are non-native to a region or to a locality or to a country. And this map shows the number of non-harmful alien species, that is invasive species at the global level. Okay, so we ask ourselves, how many species of amphibian are there in Nigeria in terms of plants, reptiles, mammals, fishes, spiders, dragonflies, and butterflies? How many species are there in Nigeria? Also, what's the nature of their distribution? If at all, we know how many species are there and their abundances. So most existing accounts are either outdated or not comprehensive, and to a greater extent, poorly sparsely documented. And thanks to Nigerian Bird Atlas project that is today accounting for the distribution and abundance of birds in Nigeria. And apart from that, I don't think if we have a comprehensive and up-to-date list about the distribution of most of our biodiversity in Nigeria. So these are some of the documented accounts of the biodiversity in Nigeria. Top left, we can see the Birds of Nigeria by Elwood, 1982. Then there is an updated version, 1994. And recently there is Birds of Nigeria by Wolfgang. And for the forest flora, we have Kennedy, 1936. We have Gazanfar, 1999, 1991 in the case of flora of Nigeria with the specific reference to a particular group of flora. Then we have one of the most comprehensive flora account in West Africa, which is Hutchison, 1954, which is almost more than half a decade, a century. Then there is also an account of flora of West Africa recently by Jones. Then there is grasses. Then there is frogs. Of course, we don't have for Nigeria, but there is a recent field guide by Channing and Rodell, 2019, for the field guide to the frogs and other amphibians of Africa, which of course covers most amphibians found in Nigeria. But again, to some extent, the distribution of some of these amphibians in Nigeria is not well accounted due to lack of knowledge about their distribution. Then for the snakes, we don't have any, but we have one specific to Old Oyo National Park by Akintunde, 2010. Then for the snakes, we don't have any, but thanks to Spalls and Branch, we have the dangerous snakes of Africa, which covers most dangerous snakes, and those mildly venomous snakes in Africa, which also covers part of Africa with the distribution maps. Then we have butterflies of West Africa by Larson, but then we are lacking a comprehensive checklist about the butterflies of Africa, but also thanks to Oscar for providing some of the list of some of the major taxonomic groups of butterflies in Africa. But then from the foregoing, you can see that most accounts about the biodiversity in Nigeria is lacking and has been long documented. So what the citizen science are doing, let's see. So we ask ourselves, why biomapping? Why do we care about biomapping? Some of us, are busy on the social media, advocating, supporting, doing biomapping. So why do we care? We do care because we need an up-to-date distribution maps, 21st century distribution maps about biodiversity. We need to track species range changes, constrictions as a result of what? Human activities, as a result of what? Climate change, as a result of drought, as a result of what? Deforestation, as a result of wildfires, as a result of so many threats out there, pollution and others. Then without biodiversity data, we cannot influence conservation policy or decision making. We need a convincing data to inform policy making. But we cannot conserve Nigeria's biodiversity effectively if we don't know where species occur. First, we need to know where they occur and what's the nature of their distribution before we can make an informed decision about species management or a particular group of organisms. Also, we need to fill huge knowledge gaps, as you have seen in the previous slides. We also need to make new discoveries because as our landscape is changing, the species account in terms of richness, in terms of distribution is also changing, either shifting or constricting. So we need to make new discoveries and rediscoveries. Also, this is very important because we are dealing with a rapidly changing landscape. Nigeria has the highest, one of the most populous country in the world. And so the environment is rapidly changing. So we need to act fast to document what's there and what's the nature of the biodiversity there. So let's look at the history of unorganized biomapping in Nigeria. Although biomapping has been done for many centuries, because it simply means taking photos of wildlife for the purpose of conservation planning and management. But in the most recent years, we are seeing an increase in unorganized biomapping activities all over the world. And that's also happening in Nigeria. So how do we arrive at where we are today? So the formal launch of the Nigeria Bird Atlas project in November 2015 largely marked the beginning of an organized citizen science in Nigeria. And the movement started with a team of 3 individuals, Dr. T, Dr. Ulf, and Dr. Sam, if I'm not mistaken. But today, we have approximately 300 citizen scientists that are contributing data in different biodiversity or citizen science projects in Nigeria. Not only the Nigerian Bird Atlas project, but also on Virtual Museum, iNaturalist, and others. And what are the attributes of this? It is as a result of atlasing and monitoring workshop in Africa, which was conducted in 2017, which aimed at promoting bird atlas and virtual museum projects in Nigeria. And from the slides there are photos, you can see the screenshot of the workshop and some of the workshop participants. And this is when we were introduced, along with other friends and other scientists in Nigeria, we were introduced to biomapping activities. So here present some of the approaches and applications to biodiversity mapping. There are so many biodiversity mapping activities out there, but for the purpose of the presentation today, I will focus only on 2, that is iNaturalist and Virtual Museum. Many out there, depending on one's preferences and goals, because they all have their cons and pros, they all have their pros and cons, and they help find out what animals are in a defined area, locality, state, country, or geographic area. Remember the definition of biodiversity mapping. It is eyeing the presence or counts of plants and animals in a given location, locality, or area or region, or at the global level. So today we can employ the use of trail cameras. We are employing the use of drones, also the use of GIS, remote sensing applications, earbird, globe, and others. So before focusing on Nigeria, it would be good if we can look at roughly the status of iNatural status in West Africa. So in West Africa, there are 3,777 observers, and to date, more than 8,500 species have been identified on and have been submitted and identified on an iNaturalist. And this consisted of over 123 observations. And interestingly, about 4 of us from Nigeria are making good attempts to do some biomapping activities on iNaturalist. You can see that Ebola, which is listed at number 5 in West Africa, with his list of observations, over 2,500, close to 2,600 observations, and that includes like 559 species. Then there is Dotun, and I'm listed as number 13, and Loki as 17. That is in West Africa. So I think this is good for us in Nigeria, despite the limited resources and some of the challenges we are all aware. I think we are incredibly doing good. So now I am focusing on Nigeria, just to give us an overview of our environment. Nigeria is located in West Africa in the Gulf of Guinea, which comprises of several rivers, forests, and of course is located on the Guinea-Congolian biodiversity hotspot, which is listed as one of the 34 biodiversity hotspots in Nigeria. And today, as of today, the population of Nigeria is over 280 million. And the landmass of Nigeria is over 920,000 square miles, which makes Nigeria as the 32nd largest country in the world. And of course, there are diverse habitat types ranging from forest to wetlands to savanna, which is also classified into different zones: Sudan savanna, Guinea savanna, and Sahel savanna. Then we also have semi-desert and desert area in the far northeast. Then, of course, we also have mountain ranges along the Obudu Plateau, Sankola mountain ranges, the Mambila Plateau, which also extends far into Cameroon. And as a result of these diverse habitat types, Nigeria has one of the high biological diversity in the world. Since the 1990s, the IUCN ranked Nigeria as 11th in terms of biodiversity richness in Africa. And among some of the great primates we have in Africa, we have 4 great primates, and we have 2. We have the chimpanzee, we have the gorilla. And in terms of birds, Nigeria is ranked 23rd in the world by BirdLife International. In terms of butterflies, Nigeria is ranked number 3 in Africa. So we have so much biodiversity richness in Nigeria. We are blessed. So what's the status of biomapping activities on iNaturalist in Nigeria? So I present the top 5 biomappers in Nigeria. So to date, we have 18,000, close to 20,000 records observations in Nigeria, and that consists of over 3,000 species, and this comprise close to 1,500 observers. So this is the coverage map of biodiversity. In Maiduguri, there is also very little activity, and this is as a result of course, some of the insurgency going on in these areas. But around Kano, Jigawa, Gombe, and central parts of the country, in the southern part, I think we are doing good in terms of mapping activities. So one interesting question we will ask ourselves is, what are the most mapped species on iNaturalist in Nigeria? The most mapped plant species is neem, which is termed invasive, and it's as invasive because it's invasive because it was introduced to many parts of the world from India. It originates from India, but introduced to many parts of the world. So it is the most biomapped plant species on iNaturalist in Nigeria. Then we have the African Mahogany, Kaya senegalensis, with 1 observation, the most biomapped mammal is straw-colored fruit bat, which is one of the most abundant bat in the world. Then in the case of reptiles, it's rainbow agama. Then for birds, it's the cattle egrets. Because some of us might think it is a Latin dog, yeah, but it's cattle egret. Then the most biomapped butterfly is small orange acre, Acre serena. Then grass is Andropogon gaianus. Then the most biomapped dragonfly is Palplura lucia. So these are the most biomapped species on iNaturalist in Nigeria. And if you're interested, you can go further to look at the distribution of this biodiversity stuff in Nigeria. You can, for the case of NIM, you can see the distribution almost all over Nigeria, even though we don't have records around the Mambila Plateau, but also in Maiduguri, it is as a result most likely of very low biomapping activities. Then for the straw-colored fruit bat, you can see the distribution, in terms of submissions. And if you click on any record, you can see some of the records, the locality, the dates, and the scientific name and the common name. Also, something of interest, we can look at the seasonality reporting for an individual submission or species. This is hooded vulture, which is a critically endangered species in Nigeria. So far, there are 20 observations from Nigeria, and this is the seasonality reporting for the hooded vulture in Nigeria. And you can see the submission, the uploads, that is the biomapping activity for the hooded vulture. And this is Latestia longicaudata. And as far as we are aware and we know, this is the first wild picture of this species from Nigeria, and if I'm not mistaken, from entire West Africa. I took the photograph of this Latestia, which is of course a lizard, in 2018 in around Kiawa. And to my surprise, when I uploaded on iNaturalist, this guy contacted me last Saturday. Today, it is the German Society for Herpetology and Herpetoculture. So they were interested in basing the distribution and abundance of lizards at the global level. So they were interested to curate this record into their database. So they sought for my permission, and I gave them the permission, and it is there on their database. You can see this is a screenshot of their record from their database. So let's look at the virtual museum. So there are so many projects under the virtual museum, just like the iNaturalist, which also covers most of virtually every taxa. So this is a project hosted by the Fitzpatrick Institute of African Ornithology in Southern Africa. Most of us are aware of it because of the Bird Lassa and the Nigerian Bird Atlas project we are involved. So there are so many projects. The BOP, for instance, is the birds with odd plumage, like birds with aberration or albinism or something. Or fish map, distribution map for fish, reptile map, distribution map for reptile, less wing, leopy map, tree map, spider map, and bed pigs, distribution map for bed pigs. So we'll take a look at some of these projects. So it's the leading and largest indigenous biomapping database on the African continent. We know that iNaturalist, it's at the global level, and it is owned by some international communities, mostly in the United States. But this is one of the leading and largest indigenous biomapping database on the African continent. And the aim is to contribute to biodiversity projects, generate distribution maps for biodiversity to serve as conservation and education tools, because we can use the biodiversity maps to influence policy. We can use the biodiversity maps to make informed conservation management planning, and we can also use it for education purposes. So this is the number of species mapped across VM projects, virtual museum projects in Nigeria. But you should please note that only projects with more than 40 species are reported here. So in the case of bed pigs, there are 399 bird species on bird pigs project submitted or mapped from Nigeria. So if you are talking about the number of bird species we have on the Nigerian Bird Atlas project, maybe we're talking about like 960 species or 40 or 60 or thereabout. So virtually we are saying that we have biomapped, we have seen almost half of the Nigerian birds. We have their pictures, we have their photos. And in the case of Lepi map, we have 319. Then in the case of Ordinator map, dragonflies and damselflies, we have 142. Reptile map, we have 51. And mammal map, we have 40. Then in other projects, we have less than 40 species mapped. So this is the number and records of DM observers on the virtual museum. But please note, this was downloaded 2 months ago. So I haven't updated the number of records in the past 2 months. So, but more or less, the number of records is more or less the same now. So these are the total number of observers on INAT on virtual museum from Nigeria. You can see, considering the number of citizen scientists we have and the number of our population in Nigeria with over 200 million people, but we have less than 30 or 40 people that are contributing data to biodiversity mapping in Nigeria as per virtual museum, which calls for an increased awareness about biodiversity mapping and biodiversity conservation awareness in Nigeria. This is the coverage map for bird pigs in Nigeria. This was also downloaded on August 2022, and you can see the number of records and the grid cells. So the squares you are seeing are grid cells, which are similar to like pentads, but this one are, if I'm not mistaken, like 25 by 25 kilometer grid cells, which are larger than pentad scales. And you can look at the number of species, the species composition in each grid cells. And this is the species richness on the bird pigs project from Nigeria. You can see the grid cells, and the numbers you are seeing are actually the number of bird species in each grid cells. You can see the grid cell around Aplori, we have 94, and that means there are 94 bird species. In terms of photos, there are 94 bird species biomapped in that grid cells. Then we have 90 around Duze, that is the grid cell in my university. We have 90 species of bird species, 90 species of birds that we biomapped. Then around Hadejongru wetlands, you can see 64. Then also around Kano, we can see 54. Then around Lapia and other areas, you can see some good numbers. And if you look well, I think there are some areas with 0. I can't see any now, but sometimes you can see 0, and that means there are records, but the records have not been identified to species level. So the number will indicate 0. Then we have some grid cells with only a single record, 1. Then this is one interesting record on BOP project, that is birds without plumage. Remember, this is a project meant specifically for birds with an aberration, maybe like deformities. And this is a long-tailed glossy starling. And for those of us that are into breeding, we know that this is not the usual long-tailed glossy starling we used to see in the field. And this was published in 2019 as a leucistic long-tailed glossy starling in the journal Malimbus. And there were about 7 reports of leucism in starlings, but apart from this record, there was no report or account of leucism in long-tailed glossy starling, and this is the first record of leucism in long-tailed glossy starling. And For the Map project, which is also one flagship projects we are all involved as citizen scientists, that is the distribution map for butterflies and moths. This is the species richness as of yesterday on the virtual museum. And you can see some good numbers of species richness around some areas around the grid cell around Lifia. And that is interesting because Atabo did his projects about biodiversity mapping in the University of Lifia, I think, and he did submit some incredible records. And thanks to that, we have about 91 species of butterflies mapped in that grid cell. Then in another grid cell around in the south, you can see there are 91 records. Then you can see some areas with 50, 72, 45, and you can go down to the grid cell level to look at the number of species and also the number of records and the last date recorded as you deem fit. And another interesting project is the Ordinator map, the distribution map for dragonflies and damselflies. And this is the coverage map for the project. The blue squares or grid cells are historical records. That is before the project was started in 2010. So there were photographic records that were uploaded on the virtual museum, specifically on the Ordinator map. So the green are the virtual museum records when the project started in 2010 onwards. And this is the species richness map for the project. You can also go into details and look at the number of QDCS, that is quarter degree grid cells, and the number of records submitted in each grid cell. So the blue is the number of grid cells, and the orange is the number of species. So in this, for the Palpleola lucia, which is the most submitted or the most biomapped dragonfly in Nigeria. And interestingly, if you remember well, this was also the most submitted record on iNaturalist, the most submitted or the most biomapped dragonfly on iNaturalist from Nigeria. At the same time, under the virtual museum, it is the most biomapped. So there are 121 records which have been submitted across 11 grid cells. In the case of this species, recorded or reported from 15 grid cells, and there are 76 records up to. So these are the most abundant species recorded on the Ordinator map. You can also go into to look at the number of species for each family, and you can see the family Libellulidae has the most number of species, almost 67 or 68 or thereabout. Then we have Agiolestidae with a single or 2 records. And these are some of the photographic records of some of the butterflies on the Ordinator map. And these are 30 species that were recorded only once on the virtual museum, that is specifically for the Ordinator map, across a single grid cell. They were recorded only once and also across a single grid cell. And you can see the last date recorded. Some like the tribe schema as far back as 2017. Then we have some recorded recently, that is in the last decade. They were recorded only recently, that is in 2019 or in 2020. Even this Pseudagrions of Lactium was recorded only recently in 2022. And these are some of the habitats that we have been doing some of these biomapping activities. You can see the types of diverse habitat types I presented earlier to you in the introductory parts. The diverse habitat types we have in Nigeria, you can see the bottom right is oasis around the far northeast, where we have the semi-desert and desert environment. You can see the wetland, you can see forest, you can see rocky outcrops and some streams, rivers. You can see mountain ranges. You can see the temperate climate around the Taraba-Mambila Plateau. And you can see very dry environment with the savanna. Yeah, so honestly, there are some incredible successes over the years as per biodiversity mapping activities in Nigeria, but there are also challenges. And one of the main challenge, as far as I know, is nothing great was ever achieved without enthusiasm. So it's enthusiasm, the ability to keep with these activities. So we have seen a number of citizen scientists that started some of these activities, not only biomapping activities, but they came on board on the Nigerian Bird Atlas project, but along the way, you know, they stopped. Even among the most nature enthusiasts, you know, they find it very difficult to submit some of these reports, I know, but I think it's lack of enthusiasm. And lack of funding is also a challenge, but that excuse only goes so far, because even around your backyard, in your home, you can do biomapping activities. There are creatures in and outside our houses, spiders, geckos, lizards, birds. You can put a bird feeder, and you can make a very good list of biomapping activities that you can also use for bird loss. You can also record and submit on the NIBAP database. Also, in the most recent years, we are faced with unprecedented security challenges, which is making it very, very difficult for most of us to go to the bush to do some biomapping activities, like kidnappings, like banditry, especially for the fact that we use some of these instruments like binoculars, like camera, which pose security challenges to security personnel and the local people. So what are the common messages today? Biodiversity data is increasingly building up on the various biomapping databases we have seen, iNaturalist and Virtual Museum, and this is creating an up-to-date abundance and distribution map of biodiversity. And over time, we can generate a national checklist for Nigeria. And as we do that, we are creating environmental consciousness, knowledge sharing, and also improve taxonomic skills. Because when you map a species and submit on iNaturalist or Virtual Museum, sometimes some of these, because taxonomy is dynamic, you know, especially in recent years where we have DNA analysis that we can go into details to separate species that were long thought as one species. So we improve our taxonomic skills. Also, we can use data, ultimately we can use this data to conserve our biodiversity or to make conservation planning and management in our protected areas or in our important biodiversity areas or in our freshwater key biodiversity areas. And the policy makers and the decision makers, they can use this data to make an informed decision about biodiversity mapping. And we can use this data as education tool, and we can use this data to develop a red list for Nigeria, which is lacking. And a lot of countries in Africa have their national red list a book, but we don't have any in Nigeria. So finally, effective species conservation is far from possible if we don't know where species occur and the nature of their distribution, but also their abundances in an environment where change is happening in a rapid pace in Nigeria. So we all ask ourselves, what is our individual contribution towards mapping Nigeria's biodiversity? And I acknowledge Aplori for inviting me to the first workshop in Africa on biodiversity, on atlasing and monitoring, which shaped my understanding about biomapping activities. The Fitzpatrick Institute of African Ontology database, the Virtual Museum, and the Nigerian Bird Atlas project. Some of these observation records were done whenever we go for Atlas activities. And the BDI, they have also been playing a good role towards some of these databasing, and they have been helping with some of these maps. And my university for allowing me to make some of these many trips at regular pace when we go out to the field, and the NCF also they have been a good collaborators. Thank you all, and I welcome your questions and comments or observations.
Thank you so much, Mr. Ringim, for this very educative and challenging presentation. You know, the truth is, we can't go anywhere unless we work together. And more of this is calling on all of us as citizen scientists It's not just enough for us to go out and keep the data in our phones, in our notebooks. It's good that we share it out there. That's only when it will be able to affect the policy or to shape the type of policy that is being drawn for Nigeria concerning the environment. And it's able to put us out there on the world map. So thank you so much, Mr. Ringim, for this presentation. And yeah,
thank you. Pleasure to be here.
Yeah. So just like he has said, please, if we have questions, we can actually just direct it to Mr. Ringim, or you can just type it in the chat, then I will read it out for him. Thank you so much. So, okay. Okay, Mr. Tunde, Olatunji, your hand is up. Please, you can go ahead with your question.
Good afternoon. Hello? Can you hear me?
Yeah, you're actually sounding very far. So if you can speak up.
Hello, can you hear me now?
Yes, sure.
Yeah.
Okay. Yeah, this is Ringim Mapping. Hello, can you hear me?
Yes, go ahead, please.
Okay. I thank you for this wonderful presentation.
A pleasure.
You really hit us with that challenges. Most of us have been having those data and we have not been populating them. Like the last time I went for up to now, I've not been able to upload all this data.
Sorry, I can't hear you well. The last time?
I said like the last time I went for Atlas in Ibadan, I think most of the data as I got there, I've not been able to upload them. I've been so busy in my place of work. But I will try as much as possible to do that.
Okay.
And in most of your presentation, I saw my name in there. This AI naturalist. That was when I was in Binigwari. I think I took some pictures of fruit bats.
Okay. Yeah.
But yes. So I saw my name there. So actually, what you said, they contacted me too. So they were like, they want to fund me, that they want me to carry out a project on those fruit bats. But I don't even have time to do all those things. So I had to call one of my colleagues in the office to carry out those projects. Dairu.
Oh, okay.
Yeah.
It will be great.
Thank you very much.
Yeah, welcome. A pleasure.
Okay, so do we have any more observation, contribution, or question from Mr. Ringim?
So it seems like it is very clear.
Yeah, obviously it's looking like that. It's very clear, and I just hope that the take-home message has sunk in very well, because the truth is, even still, I would direct this to Mr. Olatunji. Please let me just briefly say this, because the thing is, you've like registered for the Home Painted Challenge, and I can see that you've been out there sharing beautiful pictures, but then you are still yet to share your data. So please, I will encourage you, if you can kindly do that, it will have better use when it's out there and it's used for scientific purposes than just sitting down there. So please, let's make out time to submit some of these very important data sets that we're having. Yeah, so please, Rose?
Yeah.
Okay.
Yeah, I think there is a comment from Atabo.
Okay.
Yeah, if you check the comment box. And
right.
Also somebody.
Okay, Mr. Tabo is saying, I think going forward, there is a need to be, there's a need for training for enthusiasts. Yeah, sure. That can be on a collaborative ground. I don't know, but we can all work together. In fact, that is one of the reasons why this call is up and we are joining, so that when you see an interesting work, you can actually key into it. We're all here to collaborate and to work and to make sure that we save our biodiversity, which is fast diminishing. So let me just stop here. Mr. Dixon, your hand is up. Please go ahead with your question. Mr. Dixon?
Yeah, I think if you don't have a question, Joe says they have made 3 observations of leucism in long-tailed glossy starling in the last 2 months around the Hadejong wetlands. And that is very, very interesting. But I don't know, were you able to get some photos of the individuals?
Yes, there are photos, just not very good photos, but then there are photos, including a one with a pink bill. So it's interesting.
Wow.
Yeah.
Wow.
Yeah,
That's
so it
great.
would be good, Mr. Joe, if you would just share it on the virtual museum so that we can all have a look, or you're planning to publish it?
Yeah, you'll soon you'll soon see it.
Okay.
Yeah. Okay, thank you. Yeah, and it will be very, very great if you can document that. It will contribute to science, to what we know about leucism, because my observation was made in 2019, and this is like roughly after 3 years, then you observed about 2 individuals. So that is like kind of interesting, because like what's happening to the long-tailed glossy starlings? Why are they going, or what's happening to their genetic makeup? It's something of interest to think of.
Yeah, sure.
Yeah.
So Mr. Elisha is saying thank you for your presentation, and that you have just motivated him to upload most of his pictures to the virtual museum. So please, okay, Mr. Elisha, kindly go ahead and do that. It would be lovely to see that. Yeah, so from Dr. Sam, nice to hear from you. And he's saying, any encouraging words for those who might want to contribute but feel like they need great cameras?
Yeah, like I said, most of us complain about funding, but that's not an excuse to some extent, because even if you don't have a sophisticated camera, you can actually do some biomapping just from your backyard. If you can put like a bird feeder, you can take some good photos, because I know Atabo, he doesn't have any sophisticated camera, but he has some of the most submitted records on some of these biodiversity databases. And he uses his camera phone, I think, if I'm not mistaken.
Yeah, okay. Lucky is
yeah,
on this
but
call.
and specifically for you, Dr. Sam, I don't think that is an excuse now that you are in the US. Yeah.
So
yeah,
Mr.
Alicia,
Lucky
please
is on
do
the
that.
call.
Submit your records. Yeah, I know you do. You do a lot of biodiversity mapping activities. Yeah, please do submit your records on both platforms. They all have their advantages and disadvantages, and we can look at that in future presentation. It is good you have your records on both platforms, because these are like database. They can fail, but if you have, it's just like a backup, and you can do different things. You can play with them differently. So it is advisable that you have your records on both platforms. For me, I use both platforms and I play with them.
Okay,
yeah.
Interesting. I'm very sure Lucky was like, his phone. Yeah, it can be challenging with a phone, but Lucky, you're doing a very good job with that.
Yeah, sure, it's very good job.
Yeah, so we don't need to have a big camera for us to go out there and do. But for most of us that are actually doing birds, we wish we have a camera.
Actually, you only need a very good or sophisticated camera if you want to go into professional wildlife photography.
Yeah. Okay. So we can actually just start with something as small as we can get.
Yes. Yeah.
So
sure.
No excuse for anybody.
Yeah. No excuse for anybody.
All right.
Yeah. Maybe the neighbor can look into maybe buying some and give some, maybe pay instrumentally, or I don't know, or give some subsidy.
Interesting. That will be forwarded to the neighbor management. Maybe they will look into it.
Yeah, but honestly, we need to document what is there in Nigeria. Nobody will come and do it for us, and we are the generation of citizen scientists that we can make history in our. This is the best we can do in our lifetime.
Yeah, sure. Yeah, thank you, Mr. Joe. I know. Yeah, there's no excuse for anybody, including me.
Yeah,
so we'll try. Even if it's just the grasses, we'll snap and put it out there. Nigeria needs to be represented. Thank you so much for directing the challenge personally to me. Okay. So let me just quickly go into the chat and then. So Ibrahim Dan Azumi is having a question for you, Mr. Ringim. So he's saying, why do virtual museum delay one submission, especially when not identified?
Yeah, so you see, honestly, identifying some of these records is incredibly challenging because there are, for most of the projects, there are one or very few individuals who dedicate their time because they are not being paid. It's voluntarily, just a volunteer, just like us citizen scientists. So they dedicate their time to identify some of these records. So whenever maybe one of them is in the field, so definitely if you submit a record, you will have a delay in identification. But as soon as they are available, so you will see most of your records are identified.
Okay.
Yeah,
so there's always a
it's
way
incredibly
out of
challenging. Yeah, but the good thing is that sooner or later, the records will be identified and they count towards biodiversity conservation.
Okay. Yeah. Thank you so much, Mr. Ringim. There's another one coming up. That some species are deceptively similar across ranges. So time is needed for cross-check. I'm very sure Atabo is saying that to back up what you are just saying to Azumi. Yeah, so the people actually doing the work of identification is not easy. So be patient with them.
Yeah, that's true. And for some species, it is very, very tricky to identify them to species level, especially in the case of butterflies, in the case of dragonflies. Some we require to have a live specimen or a dead specimen to take a closer look of the specimen before you can identify species to species level. So because it is a photographic record. So that is why for some species, they will be there for years, even decades, without being identified to species level. Yeah, sure. Because there are some really, really tricky genus, just like birds, like the cisticolors or something, it's very, very hard to identify them to species level. And considering also the camera challenge, some cameras are very poor, so the pictures are not clear or they are very tiny. If you zoom, they look blurred, so you can't make any meaningful identification to species level. So it's better left at genus level. Yeah, sure. Yeah, but I think now that we have a DNA lab, I don't know that it is start functioning in uploading. So we can, as time goes on, when we go to the field, we will be doing some collections and we can be identifying some of these records in the uploading lab. So I think we are moving in the right direction. Yeah, sure. The lab is functional. Yes. And so it's very much open for collaboration. Yeah. So yeah. So thank you so much all for joining. It is a pleasure to be with you. Thank you so much, Mr. Ringim, for this wonderful presentation. It's a pleasure to be
Yeah, and for all that join in the call today. We thank you for creating time to join us. And
thank you.
yeah, so just a quick notification, this call is a monthly call, right, from organized by the Nigerian Bird Atlas Project. So it would be nice that we all create time to join in from time to time. As you can see from today's presentation, it's really, really educative. And it will be very good to have all of us carry along so that we'll all receive the challenge, we'll all hold our hands and move forward so that we'll be able to save the biodiversity that we have in our dear nation, Nigeria. So thank you so much again for joining, and sure looking forward to seeing all of us joining the next call, which will be very soon in the second week probably of November. So thank you, and have a very good evening, or some in other parts of the world, have a very good morning or afternoon. Thank you so much. Thank you again, Mr. Ringim. So
it's a pleasure to be with you.
if you can unmute your cameras. I know we have challenges with internet, but if you can unmute your cameras and just wave a bye, it will be nice to see your faces. Thank you so much.
Bye-bye. Okay. Bye-bye.
All right.
Good to see you.
Bye.
Yeah, bye.
Thank you.